Hello everybody and welcome to another episode of Medisodes. I'm Adrian and here with me are Anupam, Surya and Shrey. This week we're going to be talking about bioprinting, what it is and its potential uses in medicine. Bioprinting is a novel technology, so it's not as well known as other technologies, but it has huge potential to benefit industries such as medicine, cosmetics and food. So we start with the questions, what is bioprinting and how does it work? Bioprinting is basically a form of 3D printing, but instead of materials that you might traditionally associate with 3D printing, such as PLA, resin, and nylon, biomaterials are used. A 3D printer is able to provide depth to what it's printing, and a bioprinter works in a similar fashion, distributing biomaterials like living cells, synthetic glue, and collagen scaffolds in layers to create an object. This process is called additive manufacturing, for the materials fed into the printer are solidified as they come out to create a 3D structure. However, in order to get to the additive manufacturing stage, the printer needs to receive a blueprint, which is a computerized image of what it's trying to create. Then the materials you want to use are fed into the printer. Note that you might not want the whole structure to be made from just a single material, so it is crucial that materials are fed into the printer at the right timings. As each layer is printed, it will cool and stick to the previous layer due to collagen, glue, or just the type of cells being used. 3D bioprinting follows three general steps: pre-bioprinting, bioprinting, and post-bioprinting. Pre-bioprinting is the process of creating a model that the printer will later create and choosing the types of material that is to be used. Common scanning technologies used for the production of model are computerized tomography and magnetic resonance imaging, otherwise known as CT and MRI respectively. To print layer by layer, tomographic reconstruction is done to the images, which creates 2D images of each layer, which are then sent to the printer. Once the image is created, the specific types of cells that are to be used are isolated and multiplied and mixed in a broth that provides oxygen and other nutrients to keep the cells alive. The next stage is bioprinting. The liquid mixture of the cells and nutrients, called bioinks, is put into a printer cartridge and deposited according to 2D scans. 3D bioprinting typically dispenses cells into a biological scaffold by a layered approach. Next is post-bioprinting, which is a stage necessary to stabilize the structure that has just been bioprinted. If this process is not done properly, the structural integrity of the 3D object will not be maintained properly. In order to maintain the bioprinted structure, chemical stimulations are needed that, for example, control the remodeling and growth of tissues, which allows for the maturation of the printed structure to be controlled. Now, let's consider the different approaches to bioprinting. Researchers in this field have produced different methods to produce living structures that are constructed with the appropriate biological and mechanical properties. Biomimicry is the first of these three approaches. The objective of this approach is to produce structures that are identical to the natural structure of what is being replicated. This involves certain factors, some of which are the shape, framework, and the biological makeup of what's being replicated. This approach, in order to be successful, requires for the tissues to be replicated on cellular levels, and a thorough understanding of the structure being replicated is crucial. Another approach to bioprinting is autonomous self-assembly. This approach is dependent on the process of organ development in embryos to be able to replicate the structures of interest. This relies on the cell to drive the differentiation and specialization of cells to the correct types and numbers to build the organ independently, and requires a deeper understanding of how embryonic tissues develop into a sophisticated structure. A third approach takes the best of both worlds and is called mini-tissue. Tissues are built as very small components, which are then assembled to form the larger complete structure. So with that, we have covered the basics of what bioprinting is and how it's implemented. Now on to Shrey, who will discuss the practical uses of bioprinting.
One of the major hopes for bioprinting is the ability to make new internal organs for those that need transplants. Currently, hundreds of thousands of people are on transplant waiting lists, and if we can manage to bioprint hearts, lungs, livers, we can reduce the time these people spend in hospital or on dialysis. And this will greatly increase patient health and well-being. The major challenges with creating an internal organ like the heart is the fact there are so many different tissues within the heart, such as connective tissue, muscle tissue, and nerves. And being able to bioprint all these different types of cells is very challenging. Recently, a team in Israel has managed to bioprint a heart. However, this is only the size of a rabbit's heart, which is incredibly small even compared to a human heart. And this shows the challenge that researchers face in trying to scale up this technology into humans. But if we were to successfully be able to bioprint an internal organ like the heart, this would be game changing to the way that we treat people with organ failures. Instead of having to wait years on a transplant list, we could send samples of their cells to a lab and then have that organ made within a couple of months. And since the biomaterials come from the patient themselves, the risk of rejection is close to zero. Currently, bioprinting has most successfully been used to make our largest organ, our skin. Currently, bioprinted skin grafts are used instead of traditional ones, and these had better outcomes for patients. Bioprinting is not only useful in transplants, but also in other medical settings.
So one of the uses of bioprinting comes in the preclinical testing of drugs. So to provide you with a bit more context, the cost of developing drugs has increased over the years significantly. So in 2018, in order to bring a drug to the market, it took approximately $2 billion and over 12 years of development. And drug approvals are also decreasing as well. Looking at the long period of time to develop a drug, this is mainly due to the failure of drugs when tested in clinical trials. This is due to a lack of accurate data about efficacy and side effects being obtained from preclinical trials. During these preclinical trials, the drugs are tested on tissues that are grown in a culture medium. These preclinical tests are therefore unrepresentative of how the tissues would actually respond to the drug, because there are many other conditions, processes, reactions, and organ systems that are found in the human body which might affect the efficacy of the drug. Animal testing, for example on mice, is also not representative because animals have different DNAs compared to humans. Bioprinting can be used to create a chip similar to a computer chip that mimics organs and their functions. These are known as organs on a chip. For example, if the effect of a drug on the lungs was to be investigated, researchers could create a chip that mimics the function of the alveoli in the lungs. This is done through using a partially permeable membrane and growing lung tissue on one side and capillary tissue on the other side. Vacuum channels can also be used to mimic breathing. This organ-on-a-chip engineering also ensures that these organ models respond to surrounding stimuli, for example infection from bacteria, making it more representative of the human body. We can develop more accurate organ models by using bioprinting combined with bioengineering. Creating multiple organ systems and linking them together to create an entire body system on one single chip can mean that we can test the effect of a drug on the entire body. This means that the effect of a drug on humans can be accurately found in preclinical trials, reducing unexpected side effects and efficacy problems in the clinical trials phase. This can save both time and money during drug development. Bioprinting can also mean that huge advances in cancer research can be made. Traditional tumor models used for testing do not represent the complex three-dimensional interactions that could occur in the human body. Again, scientists understand that building more accurate models of tumors can mean that their pathology and response to treatments can be observed reliably before being used on humans. To ensure that organ systems of both uses mentioned work effectively, bioprinting must make sure that the organs respond to stimuli and the environment and change their structure over time. This is known as 4D bioprinting. To summarize, bioprinting is a great method of collecting more accurate data in research trials while also saving time and money. Now on to Anupam for the ethics behind bioprinting.
So there are many different problems when it comes to bioprinting in terms of how it's going to be implemented in our society. So I think the first one we're going to discuss is, where do you get the raw materials from bioprinting? Already, people have been discussing stem cells as a source, and while in most cases these can be harvested from the adults themselves, sometimes they have to be harvested from embryos or umbilical cords. What do you think, guys? Do you think it's ethical to be harvesting embryos from other people to use in this 3D printing?
I think it's not ideal in a perfect world, we'd be able to use stem cells from the patient because that also reduces the risk of rejection from the body, which is another major problem with organ transplants, is the fact that organs get rejected. And that means that a healthy organ is wasted, basically.
Yes, the reason you have to use in certain situations embryonic stem cells is because those stem cells are what we call totipotent. So that means they have the ability to differentiate into any type of body cell. A lot of the time, the stem cells harvested from adults are pluripotent, which means they have a limited number of cells they can specialize in. And while this works fine if you're trying to create something such as bone marrow, which is already happening in certain places around the world, it becomes a lot more difficult when you're trying to replicate larger organs. And in those situations, it's a lot easier to use these totipotent cells. Another problem this sort of leads on to is safety. How safe are these 3D printed organs when put in people? Shrey's already talked about the issue of rejection when it comes to being given a transplant. And there are theories that even worse problems could occur from putting artificial organs into humans, especially ones that come from a 3D printer. So what do you guys think about that sort of risk?
Some types of risks from 3D printing are inevitable, such as like contamination, because you're inserting it into someone in the case of organ transplants. And those sterile environments do exist, and radiation treatments. It's really hard to control the safety of 3D printing, and especially the materials that are used. It's really easy for contamination, which could cause even worse problems in the patient.
I'd like to point out, though, that many people on transplant lists have failing hearts, and surely even the risk of contamination, surely that's better than the inevitable failing of a heart and being put on life support.
So Surya, what do you think?
So I think it's really important to understand that there are two sides to this, where people who need organ donations need to receive their organs, but it's also about balancing the health and safety concerns of bioprinting as well, because there is a risk of infection, as Adrian said. So I think it's about finding the balance between the two that we need to achieve.
I would like to turn this notion of safety on its head a bit, because as Shree was talking about, bioprinting can have a really important role within clinical trials. And one drug might work in a certain person, but have devastating effects in another patient. And I believe that this organ on a chip technology can be used to create personalized medicine, that you can check that a medicine will work in their body before you actually administer it to them. And that could improve patient safety.
That's a very good point. It does mean that if we are able to produce organs 3D printed just on demand, human testing might become a thing of the past. Right now, there's hundreds of clinical trials going on for humans for the COVID-19 vaccine. But maybe in the future, we won't need to test the COVID-19 vaccine on real humans because we can test it in simulated humans almost using 3D printed organs. So if you look at it that way, 3D printed organs are a huge step forward.
It would also reduce animal testing, which is another big ethical issue in the development of drugs. Many people oppose the use of animals and making animals suffer. So if we could have bioengineered organs to test on, we would need less animals in preclinical testing.
It is a massive step forward in terms of our scientific capabilities. However, the technology, as we've said, is very new. And that brings up another ethical question. Who's regulating all of this? Currently, only two countries, South Korea and Japan, have had regulation put out by their medical device regulation administrations. No other countries have been able to put out definitive guidelines. The FDA is stuck on decision whether to categorize these bioprinters as biologics or devices and what sort of rules they'll fall under. And there are further concerns about intellectual property and ownership for organ designs. What if one day your organs get copied and shared across the internet? What do you guys think about the risks that poses, especially without good regulation?
I mean, I certainly wouldn't accept bioprinting in medicine without decent regulation. Even with regulation, there are loopholes that people try and sneak through. Without regulation, the safety of patients is at risk. And as medical professionals, doctors can't ever risk the safety of their patients.
So do you think that in the future there will be regulation that can allow 3D printing of organs to happen?
I think similar to how organ transplants have to be heavily regulated, and all medical practices have to be regulated because of the fact that you're dealing with people's health. It's the most important thing to a population, and without strict controls on what companies can do, should the private sector even be allowed to do this? That's another question. Should it be left to them, or should it be left to universities and national health services? I think whenever, especially something as personal as your own organs, I think there needs to be really strong regulation.
Now, a further question about this is, while it may be feasible to see in the future countries such as the United Kingdom or the United States put in place regulations on organ transplants, there are other countries where regulations already aren't upheld around ethical issues such as this. Do you think we could see a black market organ trade develop because of this 3D printing technology?
I mean, I certainly think it's a possibility, and a scary one at that. Because when organs are being printed cheaply, corners are being cut, safety measures are not being followed, and this obviously risks the life of the patient. And I think to prevent a black market, there's going to be things such as serial numbers being allocated to officially 3D printed organs, and this increases the cost of the technology even more. So I think we're a long while off this being a commonplace thing, but it is certainly a possibility.
Now, Adrian, you made a good point there about cost of all of this. Now, again, we've been talking about how wonderful this technology is, but at the end of the day, it's going to cost someone. For example, the National Health Service is already struggling to pay for a lot of its services. So maybe in the UK, it might end up becoming a case of only the rich can afford it. Do you think that that's right, that this sort of technology should only be available to the rich?
I think this technology is going to be used to fill a gap that's already present within the NHS. Thousands of people in the UK are on organ transplant waiting lists, and most of them won't survive to see the organs that they need. And ultimately, the NHS has a duty to do whatever it can to increase the chance of survival of its patients. And if bioprinting can help that, I think the NHS should do it.
Again, this is creating an economic divide, and this compromises the justice of the medical system. And it's really important to understand that medical resources should be distributed evenly across the population. And if only a certain percentage of the population who are wealthy obtain this technology, it is not ethically justified to make this available in the free market.
That's a very good point. And let's expand that. So maybe not just within a country, but across the world. This is already actually happening with the COVID vaccines. Countries such as the UK and the USA are going to be delivering these vaccines to their people a lot earlier than countries, say, in Africa. Do you think that a similar thing happening with this technology is right?
I don't think it's a question of right or wrong, because obviously it's wrong that some people have access to healthcare and life-saving measures earlier than others. But I think it's inevitable, considering that all things come with a cost, and someone has to pay for it. And those that are more able to pay for it will get it first. And that's the sad truth of it.
That is indeed quite a sad truth. Now, we've been talking about some quite serious ethical issues, so let's lighten up a bit with this next one. So obviously, when you're 3D printing organs, part of the technological advantage is that they can be custom made for the person. And while they can be made to be almost human or exactly like human organs, what's to stop scientists from making these organs better than current human organs? Now, I'm not talking about giving people super strength or super speed, but increased lung capacity is a very possible achievement from this technology, and it can have a massive impact on how a person exists. Do you think that this sort of technology should be used in this way to enhance humans beyond their natural capabilities?
Carry on with the superhero theme that's going. As a great man once said, with great power comes great responsibility. And I think this applies to bioprinting as well as many other new technologies in medicine. We can do so much, but whether we choose to do that is another question.
I certainly don't think that is ethical to enhance humans with bioprinting and organs, because then if we link back to the issue of money that we had, the rich would be able to enhance themselves more than those who couldn't afford it. And that's another ethical issue, and it's unfair on those who can't afford it. And another thing that could be linked to this is sports. I mean, currently, performance enhancing drugs are illegal, but how would you control this?
That's a good point that you've made, that it will give the rich people who can afford this enhancement an advantage over other people. But here's a question. Firefighters spend a lot of their times going into burning buildings. Would it not help firefighters to have these enhancements so they can save lives more often? It's not as cut and dry, I think, as saying we shouldn't enhance people beyond the normal level.
Well, I believe that this technology should only be used on those who absolutely need it. I mean, enhancing humans is all well and good, but the purpose of most medical technologies is to help people who are already sick rather than just improving on people who are healthy. And we should prioritize helping the sick rather than trying to enhance those that are already healthy.
Again, very good point. But look at it in this sort of context, I think. I'm just playing devil's advocate here. Say a person with asthma develops a lung tumor and then they need a lung transplant. When you transplant the new lung into them, do you also cure their asthma at the same time using an enhanced organ? Or do you restore them to how they were before?
I think it would be wrong to change a person like that. And I think it's similar to the problems with genetic engineering. I don't think we should be interfering with how people are. And I think that's a very risky thing to do and could have unintentional consequences that may harm the patient.
So the NHS has limited resources, and though we would love to cure everything about the patient and make them as healthy as possible, this is not practically possible due to the fact that the NHS is already struggling with its resources, and bioprinting is not infinite. So we need to consider that as well.
All right, guys, so I think we've covered bioprinting and its basics. It's a novel technology that's got a promising future. Tune in next week when we have another Medisodes episode. Remember to like and subscribe and comment down below. We really do appreciate it. Thank you for listening, and we'll catch you in the next one.